Hello, and welcome to today's iSNTD Connect webinar. My name is Marianne Compered, and I'm speaking from the International Society for Neglected Tropical Diseases. Today, we are going to be speaking about a neglected tropical diseases, which remains quite mysterious. The mode of transmission still eludes us, and yet can have a devastating impact on its patients. We'll be looking at Mycobacterium ulcerans, the bacteria causing the causative agent of Buruli ulcer. And joining us today from Melbourne in Australia, it's my pleasure to welcome Dr. Koen Vandelannoote. Good evening, good night, actually. Koen, how are you doing?
Yeah, hi there. Hi, everyone.
And thank you so much for joining us at what is about 10 p.m. your local time. Much appreciated. Thank you so much. Koen,
no worries.
Koen, you are a scientist working at the Doherty Institute for Infection and Immunity as part of the University of Melbourne. And today we'll be hearing a little bit more about your group's research on the resurgence of Mycobacterium ulcerans following the damming of a river. And this is a really crucial insight into modes of transmission, into the relationship between the environment and disease, and really underpins, this kind of work underpins what the new roadmap at WHO, the Neglected Tropical Disease Roadmap, up to 2030 is trying to do, really trying to bring in different disciplines, working together to understand better the way disease affects us, and also really foster those important collaborations across sector to improve their control. So, Koen, I'll hand over to you now. Thank you again for joining us today. And as usual, dear attendees, if you have any comments or questions you would like to ask Koen at the end of the presentation, please feel free to include those in writing in our chat function. And welcome to you as well, and thank you for joining us as well. Over to you, Koen, and thank you very much.
Thank you very much. As Marianne already said, I'm a computational microbiologist, and I work at the Doherty, which is very close to the University of Melbourne campus. To be complete, like the majority of the work that is described in this presentation was actually done at the Institute of Tropical Medicine in Antwerp, Belgium. So today I'll be talking to you how Mycobacterium ulcerans was introduced into the Bankim Health District of Cameroon, probably following the damming of the Mapé River. And we'll be tackling like the research questions that I've laid out in this presentation using the power of bacterial population genomics in tandem with molecular dating approaches and remote sensing analysis. So I'll first just briefly start saying a couple words about Buruli ulcer epidemiology, and from then we'll move to the bacterial population genomics, the molecular dating, and at the end I'll say a couple words about the remote sensing analysis that came in parallel with this study. So like Buruli ulcer, it's an infection of the subcutaneous tissues with the environmental pathogen Mycobacterium ulcerans. Now, in 70 years of research in Africa, there's still a very wide knowledge gap in our understanding of how Mycobacterium ulcerans is transmitted and what its environmental reservoirs are. So unlike leprosy and tuberculosis that are characterized by person-to-person transmission, Buruli ulcer is not contagious, and it's considered a non-communicable disease. So in Africa, humans are the only known reservoirs for Mycobacterium ulcerans, even though various non-human reservoirs have been put forward, for which experimental evidence currently is still lacking. In Africa, the only known vector species, in Africa, there is no known vector species, even though, again, a number have been put forward without any sound experimental evidence really backing it. So in epidemiological investigations, it was already established early on that Buruli ulcer is linked to rural wetlands, specifically those with stagnant or slow-flowing water. Now, in various reports, we can read that changes to landscape hydrology have been linked to Burley ulcer disease outbreaks, and as such, an increase in Burley ulcer incidence has been associated with both natural and human-associated disturbances like exceptional flooding, for example, after heavy rainfall, or the implementation of irrigation systems, or alluvial mining operations, but also with the creations of impoundments and wetlands. So even though these anecdotal associations between disturbed water bodies and an increase in Burley ulcer incidence is actually quite commonplace in the Burley ulcer literature, a direct evidence of such a causal association has always remained lacking. So because we'll be looking at the situation in Cameroon, I'm just going to introduce view in Cameroon very briefly. So the first Burley ulcer endemic area of Cameroon, it was described in 1969 along the Nyong River that's located between the cities of Eos and Akonolinga. And you see that little hotspot over here pretty much. However, 35 years later in 2004, there was an additional Burley ulcer endemic area that was identified in the Bankim health district of Cameroon, and that is located over here in the north of Cameroon along the border with Nigeria. So the environment around the Bankim Burley ulcer focus is dominated by Lake Bankim that you see over here. And Lake Bankim is an impoundment that was created by damming the Mapé River. So the Mapé River used to flow here, and it was dammed very close to its mouth in the Mbam River that's located here, and that created that impoundment that you see over there, which is Lake Bankim. So the Mapé storage dam project was implemented in the 1980s to meet Cameroon's electricity demands and to also regulate the flow of the Sanaga River during the dry season. So you can't really see that on that map, but downstream is a very important stream called the Sanaga, and they really wanted to make sure that it always had sufficient of water to keep it navigable. So the infrastructure works of that damming project started up in 1985, and very quickly in 1988, the structures were already finished, and the dam was completed and filled up. So there have been a number of studies that have hinted at the possible role of the Mapé storage dam project and the creation of that impoundment, of the effect that that had on the increase in incidence of Burley ulcer. So additionally, the local population that lived in that area had also suspected that the damming of the river and the creation of that impoundment has led to an increase in Burley ulcer incidence. So it's pretty hard to establish whether Burley ulcer is actually a new a new emerging disease in the region, or whether it was pre-existing and only recently recognized in 2004. And that was definitely the case, because the oral reports about disease in the region before 2004 can't really be confirmed, because there's no clinical records of any cases before 2004, and laboratory confirmation of clinical diagnosis was never sought for these potential patients that might have existed. So so this is why in this research project we aimed to use the tools of bacterial genomics in tandem with molecular dating approaches to test the hypothesis that Burley ulcer arrived in the Bankim Health District after damming the river. So I think the vast majority of you will already be familiar with the basics of bacterial population genomics. So unlike when you do a PCR and you Sanger sequence a fragment. In whole genome sequencing, you sequence the entire bacterial genome using technologies that are commercialized by Illumina, Pacific Biosciences, or Oxford Nanopore Technology. However, I'm pretty sure that bacterial molecular dating approaches are pretty obscure for most of you. So I just wanted to introduce you to the very basics, pretty much, of what molecular dating using Bayesian approaches is about. Similar to other phylogenetic approaches like maximum likelihood, for example, or neighbor joining, the goal really is to infer like a tree or a phylogeny. And this phylogeny pretty much shows us how sequences are related to each other. So in the tree, you can clearly see whether one clade is more related to another clade or to another one. Unlike other phylogenetic approaches, though, molecular dating Bayesian phylogenetic approaches, they add an extra dimension to a phylogenetic tree, namely they add a time axis. So with Bayesian molecular dating approaches like these, you pretty much can create something like a time tree, and on that time tree you can date evolutionary events of interest. So you might wonder, how is this possible? Well, first of all, at the basics of all of this is a model of molecular evolution. That is also the case in the maximum likelihood approach, for example. However, to add time within phylogenies, you need to add like some kind of time calibration, I'd call it. And in this case, this is provided by the isolation times of the heterochroneous isolate panel that we use in these analyses. So the disease isolates which we use in these analyses, they originate from different years, spanning all the way up to 50 years in the case of Mycobacterium ulcerans research. Using these approaches has been particularly successful, successfully applied to a number of studies in Mycobacterium ulcerans in Africa and in Oceania. And I've written a couple of links to those studies down there for people who want to know more. In this case, looking at the situation in Bankim, we're really interested in finding the time to most recent common ancestor of Mycobacterium ulcerans within that particular outbreak. So you can see the 40 different isolates that we use in the study. They're depicted here as black stars. Again, Lake Bankim, the Mapé River, and the Mbam River located here. So these patients, they were actually recruited between 2010 and 2012 in an exhausted cross-sectional house-by-house survey that was organized by a team of Cameroonian and Swiss researchers. So I wasn't part of that team. I downloaded the reads of their work actually from the NCBI short read archive. But this team of researchers has been involved in the subsequent part of the research project on which I've been presenting on. However, in addition, we've also added another collection of reads, 157 reads of African Mycobacterium ulcerans genomes, all from lineage 1, which we included to provide appropriate genetic contexts for interpreting the diversity and the evolution of Mycobacterium ulcerans from the Mapé River basin. Now we get a little bit to the technicalities, I guess. These reads, they were actually sequenced by the Sanger Institute for that group of Cameroonian and Swiss researchers. The Sanger Institute produced Illumina reads for those isolates, and these reads, they were processed using a pipeline called Snippy, and pretty much works like taking those resets and mapping them to a bacterial reference chromosome. In this case, we used the Ghanaian closed bacterial reference chromosome, which is very related to these Cameroonian isolates. So we mapped read sets using a program called BWA, and then the read stacks that were mapped to that bacterial reference chromosome were analyzed with a variant caller called freebase, and that allowed us to identify variation like small indels or SNP polymorphisms. So any variation that we discovered in repetitive regions of the 5.6 genome, megabase genome of Mycobacterium ulcerans, those regions were discarded. And there's actually quite a bit of repetitive sequence in Mycobacterium ulcerans. So the genome is interspersed with insertion sequence elements, IS2404 and IS2606, multiple hundreds of them. So yeah, that's almost 11% of the bacterial reference chromosome of AGY99. So if we do this for all the isolates in our isolate panel, pretty much we can concatenate all the polymorphisms that we identified and create something called a multiple sequence alignment of core SNPs and small indels. When we analyzed so the diversity that we discovered using this approach, pretty much in Bankim, only in Bankim, we identified just 95 SNPs according to across the bacterial chromosome. So these 95 SNPs, imagine along 5,600,000 base pair bacterial chromosome, that's very limited. So the nucleotide divergence was very limited. If you calculate it, it's just one SNP for every 52 kilobases. And those SNPs, they also were distributed uniformly along the bacterial chromosome. So it wasn't like one specific region that was accumulating all this this variation. No, they were quite neutral and accumulating quite uniformly. So that's just looking at the Bankim outbreak isolates. However, if we look at the extended African isolate panel, we identified 4,266 SNPs. And using our molecular dating approach, pretty much we were able to infer a mean genome-wide substitution rate of 1 to the power of -6 substitutions per site per year. And you can see the highest posterior density interval next to that. So, I mean, in more human terms, I guess, this amounts to an accumulation of approximately 0.3 SNPs per bacterial chromosome per year. So you can see that the substitution rate is really very limited in Mycobacterium ulcerans. It's also very limited in closely related Mycobacterium species like Mycobacterium tuberculosis, for example. So it's really one of the slowest molecular clocks that you can find out there. So we then so there to the right, you can see the Bayesian maximum credibility time tree that we inferred from the alignment of the extended African panel, and all 40 isolates from the Mapé region. They pretty much corresponded to lineage Africa 1, and the uncommon lineage Africa 2, it was not identified in the study region. So on the time tree that you see here, you pretty much only see lineage Africa 1. If I wanted to include lineage Africa 2 isolates, I would have needed 10 of these screens to show you the complete phylogenetic tree. But since there weren't any lineage Africa 2 isolates in this little clade of Mapé isolates, didn't include it in this tree. So we really see like a very strongly supported monophyletic group in the time tree there. However, also of note is actually that, you know, the other outbreak that I referred to earlier, that outbreak actually is distinct and separate from the other Cameroonian isolates from the Mapé region. So they form a separate, distinct phylogenetic monophyletic group. So we really see when I first saw this, I thought this was quite staggering, actually, because, I mean, keep in mind that these clades, this clade is actually more related to a clade of 135 different ulcerans isolates that have been sampled all over the African continent. So you can see that clade over here. I know you can't read the labels properly, but these are the isolates that were sampled pretty much all over Western Central Africa. So as a result, we really observed distinct spatial clustering of ulcerans from the different BU foci, which indicate strong geographical clustering and geographical restrictions on the dispersal of Bruley ulcer over the years. I also referred to the fact that you can add like a time axis on a time tree, and you can see that time tree displayed on this, that time axis displayed on this time tree here at the bottom. I'm now going to focus just on that Mapé and BAM clade that I was mentioning before. Here you can see a zoomed in version of the clade. I'm just going to take a sip of water. And it's kind of remarkable, but I mean, the Bankim Health District isolates, they shared a common ancestor around the year 2000. So this is the the ancestor of all the isolates that we sequenced from the region, and you can see that that region here is the HPD interval around that. So you could look at this as a confidence interval, even though statisticians would murder me on the spot if I would say so. But for the sake of this presentation, you can look at that particular interval in that way. So this is the ancestor of ulcerans in the Bankim region. And if you remember, this little line here refers to 1985 when the dam was constructed, and this line refers to 1988 when the construction finished and the dam was filled up. And this is 2004 when the first cases were confirmed and reported around Lake Bankim. So yeah, we found some pretty compelling support for Mycobacterium ulcerans event that closely followed damming of the river. And we also see that apparently Lake Bankim has been, let's say, endemic with Mycobacterium ulcerans for at least 4 years before it was first reported in patients. So that refers to that time period before the time to most recent common ancestor and the first reported cases. So this year 2000 introduction event is actually pretty remarkably recent. I refer to other studies that we've done, and yeah, I mean, like there we really saw that clonal expansions that are associated with hydrological basins, that these always had a time to the most recent common ancestor that was a lot further back in time, pretty much. So we did similar work pretty much on the Nyong River Basin that I already mentioned in Cameroon, but also the Uhuman River Basin in Benin and the Congo River Basin in the Democratic Republic of Congo. But even like a clonal expansion of NADBIC that I studied here in southeastern Australia in Melbourne, it also dated back temporally to somewhere in the 19th and the 20th century, usually the time when these places, when they were occupied by European colonizers during the time of the scramble for Africa. So also remarkable is that these HPD intervals, that they're actually quite narrow. So in this case, it's just an 11-year period. So if I look back at the older AdBreaks that I've studied, these all had very large HPD intervals. So they for very often they included like HPD intervals of like 100 years. So yeah, that's it's pretty remarkable evidence for a recent introduction event there. So like the next step that you could then do is look at your time tree and investigate a possible origin of the Mapé BAM outbreak when compared to the genomes of the other Mapé BAM basin. No, sorry, when comparing the genomes of the Mapé BAM basin to other West and Central African buruli ulcer endemic countries. You can see the little clade here, but these are the most related species. These are the most related isolates in the study. I made a little table here that kind of follows along. So here you can see, again, the location of the Mapé BAM outbreak, and the most closely related isolates are these two here. And they're actually from an outbreak in the Kwilu in the Republic of Congo. So these are just 50 SNPs apart, even though these two locations are separated by a geographical distance of around 1,000 kilometers. And the isolates from Mapé and BAM and the Republic of Congo here, they shared an ancestor somewhere in 1971. So if we go even further, the next closely related isolates are actually from neighboring Nigeria. So you kind of would suggest that, like the opposite would actually happen, but in reality, the isolates from the Republic of Congo somehow are more related than the ones from neighboring Nigeria. So one isolate here from the Cross River state in Nigeria had a genetic difference of 73 SNPs, and it was separated from the BAM outbreak by 280 kilometers as the crow flies. And it shared an ancestor with the BAM outbreak isolates in 1943. So the same thing is true with this particular isolate that was found in the Anambra states, which is a little bit more genetically different, an extra 70 SNPs or so, and a little bit more further away. So even though I guess we found phylogenetic evidence for a possible link with isolates from the Republic of Congo and Western Nigeria, it's not really possible to specify that these places are the origin of the Mapé and BAM basin buruli ulcer focus, especially given the fact that we have limited isolates from both countries. So as a consequence, the route of introduction and the immediate origin of Mycobacterium ulcerans introduced into the Mapé and BAM basin still remains to be established. So this is only a temporal association, and a temporal association that we've made between the appearance of buruli ulcer and this emulcerans introduction event. It does not allow us to make a causal inference, pretty much. So there's lots of environmental consequences that happen when you dam a stream. So there's direct impacts to various properties of the river and its immediate environment. So there's biological impacts. For example, you can think of other diseases that are that are spread by a vector. I'm not saying that emulcerans in Africa is, but you could see, for example, snail vector and cystiomyces, how in the literature damming rivers pretty much increases the incidence there. The same thing is true for mosquito vectors in bankophthoses or in malaria. Similarly, onchocercidosis with its fly vector, there's also evidence in the literature that shows that damming river pretty much can increase the distribution of the vector. There's also chemical and physical effects. So if you pretty much create a big pool of stagnant water, it usually gets a higher temperature, a lower oxygen concentration. And I mean, the flow of the water is obviously reduced. So as a next step, we wanted to see with remote sensing imagery analysis how we could investigate how the emergence of buruli ulcer in around Lake Bankim, how it might have been a consequence of some marked changes in the environment that were caused by the filling of that impoundment. So just, I mean, very basic all we did was passive remote sensing. So there's also active remote sensing where you just beam some electromagnetic light at the Earth's surface and you collect the light that's reflected. So, but here we took advantage of just the sun sending out electromagnetic energy and objects on the surface of the Earth reflecting that, and that electromagnetic energy being captured by Landsat satellites. So there's it goes all the way up to Landsat 8, if I'm correct. So there supposedly were supposed to be 8 Landsat satellites, but I think Landsat satellite 6 never really made it to orbit. So these satellites, depending on when they were put into orbit, they pretty much take pictures at a specific spectral band, various composite images. You can see an example depicted here to the right. And yeah, and it's important to realize that each material has like its own spectral signature that you can use for a material classification. So, I mean, the way you would go about that pretty much is by selecting like a little piece of water, for example, on one of these satellite images in quantum use and saying, yep, this is water. Do the same thing from some water from the Bankim River or from the Mapé River, and just, you know, like add some extra confidence there that yes, indeed, this is water. You could do the same thing for other land cover types like forests or grassland, and then you can use algorithms to pretty much classify an entire array of pictures to analyze how differences in land cover might have been a result from damming the Mapé River. So here you can see the result of such an analysis, pretty much where we try to quantify changes in land cover use. So I already mentioned that we used Landsat Earth imaging satellites. Again, like that data I pretty much downloaded from the United States Geological Service. And I've used quantum use to analyze the satellite images using the semi-automated classification plugin that I described very briefly before. So what you're looking at here is some major environmental changes between the period before the dam was there and after it was completely filled up. So I've used a Landsat image from 1984, and I compare it here to an image of 2012. So because this automated classifier pretty much allowed me to analyze the entire image, I'm able to make some pretty accurate estimations of changes in land cover changes. So I mean, like damming the river apparently flooded 158 square kilometers of forests and 78 square kilometers of grassland and open soil. And it also created swamps and meadows along the newly created shoreline, which is about 1,000 kilometers. So in addition, in the immediate surrounding around Lake Bankim, we can also see that quite a lot of forest was cleared through slash and burn activities. And there was also a lot more built up area between those two time periods. I just want to make clear that I don't know if the changes in land cover use that they pretty much facilitated the transmission of Mycobacterium ulcerans in the region. So I've done like a similar analysis. Here I was previously I was just comparing two different pictures to each other, but I've also done the same work where I analyzed all of the satellite images that Landsat covered over the years, close to 100 images, I believe, and no, close to 50 images, yeah, and using all this image material that was observed between 1988 and 2005, I was able to make this, let's say, a composite image that looks at the hydrodynamics of the lake over time since it was created. So if you look at the image, you can see all the blue areas here. Those were actually the regions where there was always water during this entire period. So However, if you look at the black areas, that's where we never really saw like a body of water. So you could see that, you know, all the way to the red, that's the maximum extent that the lake was ever observed through satellites observation. So you can see that there's, you know, like a considerable amount of inter and intra-annual variability in the lake surface area. So if the lake goes from its maximum extent to the region where it was pretty much always observed that there was water, we can calculate that there's close to 154 square kilometers of area that can be flooded when the lake is filled. So just time for a couple quick conclusions, I reckon. So I think we provided some pretty decent evidence that the year 2000 the Common Area introduction event took place in our temporal analysis, and that closely followed damming of the river in 1988. And I think we also showed that since its introduction in the Bankim region, that the region probably had been endemic for approximately 4 years before the first patients were reported. This is very similar to the work that we were doing in Victoria and Australia as well. And, you know, based on our observations, our remote sensing, it's kind of tempting to posit that the damming of the river, that it really created those typical slow-flowing habitats that are, you know, like a really well-established risk factor of Mycobacterium ulcerans infection. So I guess, like, you know, the strongest conclusion that came out of it is that we found a strong temporal association between the creation of the dam and the emergence of BU, but yeah, unfortunately we can't like really prove causation. So I tried to push this in a 30-minute presentation, but this presentation was actually largely published work that you can find in PLOS NTD. Happy to answer some questions about the presentation that I just did. But yeah, a lot more detail can be found in that PLOS NTD publication pretty much. So with that, I'd like to acknowledge all the people that have contributed to this work. Especially like my employers, both in the Institute of Tropical Medicine and here in the Doherty Institute. I'd also like to thank people that have contributed financially, organizations that have contributed financially, the Flemish government, namely. But perhaps most importantly, I'd like to acknowledge the contribution of the Cameroonian researchers and the Swiss researchers who actually did the epidemiological investigation in the Bankim Health District that allowed me to do all of this work. I've never been to Cameroon, so I've only worked with reads that I downloaded from the SRA and from satellite images that I downloaded from the United States Geological Surveys. But thanks to their brilliant work, I was still able to pull this analysis off. So yeah, with that, I'd like to say thank you for your attention, and I'd love to answer some questions if there are any.
Well, thank you so much, Koen. And most certainly there have been lots of questions and comments, lots of thank yous as well for this wonderful presentation. Bacterial ulcer eludes scientists in many, many ways. So this incredible approach that you've shown us, Koen, really brings in a lot of new approaches and techniques, very exciting opportunities to understand a little bit better where and how this disease is progressing. For those who aren't familiar with Koen's work, this really builds on over a decade of work in this field. All the way back in 2010, you published a paper that was really reporting on the first successful application of real-time PCR for detecting Mycobacterium ulcerans in Ghana. So a huge amount of work and really using genomics to shed a bit more light onto the detail of Bacterial ulcer and the dynamics of the Mycobacterium ulcerans population. So thank you so much. A really fantastic presentation. And absolutely, you certainly have given us much food for thought and quite a few questions coming through here on the chat. So I'll first start with a question from Roland Bamu. Hello, Roland. I think you are tuning in from the University of Shang in West Cameroon. Bonjour. And so Roland was asking, was Buruli ulcer existing in the area before the dam construction? So is there any evidence of cases before the dam construction? And second question, Roland was also wondering, is it possible to find Buruli ulcer on other sites with dam constructions, and particularly referencing the Memvele hydroelectric dam that's just been operating for about a year in South Cameroon?
Yeah, thank you for the question. So I think during the presentation I referred to these oral reports. So the local population expected, I should say, some of the local population expected that there was Buruli ulcer before the dam was constructed. However, others conflicted that. But we have no way of really knowing, because there were no clinical documentation that survived it through the times, and microbiological confirmation was never sought. So we have no evidence of there being Buruli ulcer before the dam was constructed, pretty much. And with regards to your other question, the only published report that I'm aware of a hydroelectrical dam being linked to an outbreak of Buruli ulcer, the only other report was a study done in Côte d'Ivoire, so in Ivory Coast. So not really aware of any other link in Africa. There was another report that looked at the Petit Saut Dam in French Guiana, where they did see an increase in the incidence of Buruli ulcer downstream of the dam, and there they hypothesized that the construction of the dam pretty much changed the flood working of the river, and they attributed to that. So.
Thank you very much, Koen. And another question here from Evans Uhuman. Evans, you've introduced yourself as independent researcher. Please feel free to let us know which organization or university you're tuning in from. It's always really interesting to know and hear who's tuning in from where. So Evans is asking, first of all, saying thank you very much for this open-minded presentation. Is there any evidence of relationship of the introduction of Mycobacterium ulcerans in a region with mining activities? And how can such a methodology help with evaluating this?
So with regards to alluvial mining, the majority of the published work that I'm aware of, happened in the Democratic Republic of Congo, specifically the Kwango River that forms the border between the DRC and Angola. So there have been quite a lot of illegal alluvial mining going on, and we've seen outbreak of local clonal complexes that happened there. These clonal complexes, though, I've studied these in the other published work that I was referencing to, and these clonal expansions, they pretty much they were closely related to like the endemic clonal expansion that we found in the Congo River Basin. So it's that's I know in Ghana there's also a very close links, very close eye on alluvial gold mining being done and the emergence of Buruli ulcer. I guess we could use this kind of methodology to investigate this. However, I mean, this is the only case where I actually found such a very recent introduction and a very limited amount of diversity that accumulated over time. So it's the only example that I'm currently aware of it actually working. So I'm not really sure if it can be used as a monitoring tool. I think I think emulsions just evolves too slowly. The molecular clock is too slow, so it's very, very hard to use it as a monitoring tool, I believe.
Evan said, I am from Côte d'Ivoire and just graduated from the University of Nangui à Bourgois. And Evans was also adding here on the chat that some mining activities have some mining companies have started their activities in endemic Buruli ulcer regions in Côte d'Ivoire. So this has given a lot of ideas about types of research to be carried out. So thank you for that. Lots of questions, of course, about the environment. And here we have now our next question from Professor John Wallace. Professor Wallace is the director of the Millersville University Center for Environmental Sciences. So Professor Wallace starts off by saying, well done for the presentation. And if I can just beam up, there we go, the question to the room. Regarding your 2000 intro event 4 years prior to any cases, did you happen to look at any other land use disturbances in this watershed closer to this time frame beside the dam construction in 1985? For example, any significant storm events?
So I think I mentioned before that I've never been to Cameroon, but the reason why I really focused on this area, because it's a very very pronounced environmental change. It's a pretty huge change to the landscape hydraulics that you see here, like a massive hydrological body of water that was that was created pretty much. So I'm not aware of any other smaller environmental disturbances or any other natural disturbances to the landscape hydrology, such as flooding or heavy rainfall. I'm not familiar to that. This was actually a comment that I received by one of the reviewers on the publication that asked me whether I could perhaps link it to exceptional rainfall in this particular region. I wasn't able to find an association there, but yeah. The issue there is if I would just step outside and have a walk in the neighborhood, I'm pretty sure I could point out quite a bit of environmental disturbances as well. So what I like about this story is that it's a pretty pronounced change in landscape hydrology that it's hard to discuss with. It's not a particular tiny one, but of course, it's very, very possible that it was something else, of course.
Brilliant. Benjamin Makepeace from the University of Liverpool. Hello, Benjamin. Is saying, nice work. Have you done any environmental sampling in the region by PCR, etc., for M. ulcerans?
So I haven't, but the team of Cameroonian researchers and Swiss researchers has been very active in this particular region. So as another publication on this that they did, they pretty much were able to detect IS-2404 in environmental samples. And I think, the most astonishing thing was that they actually detected the presence of M. ulcerans DNA in the environment quite far away from the hydrological dam. And in their research, it seems to suggest that, M. ulcerans infection was actually more associated with the Mapé River rather than with Lake Bankim. And another very interesting part that they noticed is that they had this environment of detritus that pretty much remained positive throughout a period of 2 years. So they kept on visiting this one particular spot, and it kept being positive even though there were no more Buruli ulcer patients around. So definitely environmental M. ulcerans DNA detected in the environment. However, given the fact that it's so difficult or close to impossible to culture M. ulcerans from the environment, it's very hard to establish whether these traces of DNA that we're detecting with this very sensitive PCR, whether that's actually proof for living Mycobacterium ulcerans out there in the environment. So it might just be that there's a very focal spot where there's a lot of bacteria in a reservoir that we currently don't know about that's leaching out that DNA pretty much that can remain in the environment for quite some while. So it's very hard to interpret these results in the disease ecology, let's say, of Buruli ulcer.
Absolutely. Just kind of looking at some of the major gaps that remain in the understanding of this disease, Roland Bamu was asking, do you think mosquitoes could be a vector of M. ulcerans? In Australia, studies implicated mosquitoes as potential vectors because they found them infected with Mycobacterium ulcerans?
So I'm actually working with a team of researchers here at the University of Melbourne who are looking very closely at this. Yeah, exactly. We know that mosquitoes are part of this tandem of the native marsupial possum, humans, and mosquitoes. So it's probably interconnecting work between the three of them. So we know that the marsupial possum that it sheds emulsions in its fecal pellets, and we believe here in Australia that mosquitoes that grow up in, for example, sewage water in which these pellets accumulate, that they can be covered with emulsions on their proboscis when they take a blood meal, for example. So there's, if you go to the houses of really ulcer patients in Australia, you can actually find Cis2404 positive mosquitoes. So that really, seems to suggest that at least here in Australia, that that the mosquitoes play a very important role in transmitting the disease. Now, researchers in Africa have been trying to replicate these findings, and until now, I'm aware of 2 studies that looked at very closely at RT-PCRing hundreds and hundreds of mosquitoes, but nobody has ever been able to find any positive mosquitoes in Africa. Also, something else that's currently missing in this Australian story, of course, is that the native Australian marsupial possum doesn't exist in Africa, so it might be another interplay with another organism. Some people are looking towards rather large rodents, grass cutters, for example, that might be playing the role of the marsupial possum in Africa. So yeah, people have looked at mosquitoes but haven't been able to find any consistent experimental evidence of mosquitoes infected with emulcerans DNA, as far as I'm aware.
That's really fascinating. So watch this space. Definitely lots of question marks left there. We've got a question as well from Rachel Simmons from the University of Surrey. And hello, Rachel. Lovely to see you here. And Rachel saying, hi, Koen, really interesting presentation. Can you infer anything about the evolution of or spread of emulcerans in the area after the introduction event? Also, do you have any thoughts on whether there are unsequenced strains that exist as they are unculturable?
So with regards to the evolution of and spread of emulcerans in the isolated from Lake Bankim, I think that would be very limited, given the fact that it was just 95 SNPs that we were able to identify. In our SNP analysis, I didn't mention this, but SNP pretty much ignores small indels. So there's like a couple of small indels that actually are also there. So there's a little bit more genetic diversity that I didn't really cover during my presentation. However, there's no large insertions or deletions that we could find. So I think, most of these point mutations that have accumulated are probably just neutral. They don't. I don't think like a particular phenotype is being selected. Most of them are in the regions that some people used to refer to as junk DNA. And then, with regards to like the unculturable Mycobacterium ulcerans, I'm not aware of any other isolates that have been isolated from the region. However, I did the sequence analysis of this particular study almost 3 years ago, I reckon, so it might be that there's more material coming in. I know that the Cameroon National Program pretty much keeps a close eye on the region, so there might be a little bit more material that has since then been produced. It's very chances of culturing emulcerans from clinical samples is relatively small if you have transportation issues. So if it takes a long time for your samples to get to the lab, and if you don't immediately start culturing them, you have a very low chance of actually culturing them from patient samples. So there's definitely a big gap in what these isolate panels that I've presented on can tell us.
Thank you very much, Koen, for that answer. We're almost coming up to the end of the time for our webinar. However, we do have one, maybe two more questions, if that's okay with you, Koen.
Yeah, no worries. Keep going.
Yeah, thank you. I think they keep coming, so thank you. So a question here from Elizabeth Giamphy. Elizabeth says, thank you for your presentation. How certain are you that the strains from the dam are the same strains, is the same strain causing infection in that geographical area?
So we know that these the 40 isolates that we had, they pretty much they originated from patients who are living in the immediate vicinity of the of Lake Bankim or of the Mapé River. So we've grown up these isolates from their clinical samples. So we know that like the genomes that we're sequencing, that it's actually like from ulcerans that's infecting these patients. So since we get it from the sample material, it's very unlikely that they get infected with another strain that we're not aware of. So,
thank you. And finally, another question from Roland. So, what is the best tool for M. ulcerans detection for qPCR? 3 systems are mainly used, and samples are considered positive if positive for the whole system. Would you have any recommendations?
So yeah, I guess the best tool if you're looking for ulcerans in the environment, that was what Roland was referring to, correct? So yeah, the best tools for detecting ulcerans in the environment would definitely be real-time qPCR of the insertion sequence element IS2404. There's like a lot of copies of IS2404 in African ulcerans, and there's also quite a lot of copies of IS2606. So I would say by doing mass screening of environmental samples and using IS2404 to detect that specific target, and to then confirm that you're actually are detecting ulcerans, you can do confirmation qPCRs, including the IS2606 assay, which is a little bit less sensitive because there's a more limited number of that insertion sequence element in the bacterial chromosome, and you can go even the next mile while trying to detect the keto-reductase genes, for example, that are found on the mycolactone-producing plasmids. There's 8 copies of them, so that's another relatively sensitive assay you could use for confirmation assay. It's very important to do these confirmation assays, though, because we've seen that there's other Mycobacteria out there in the environment that also have IS2404, so it gets kind of tricky doing these kind of environmental qPCR surveys. You really want to be sure that you're detecting M. ulcerans DNA and not the DNA of some other closely related organism, which might not cause Mycobacterium ulcerans infection.
Brilliant. Well, thank you so much, Koen. This is obviously lots of questions still up in the air, still remaining. It seems to be a very tightly knit community, the one around Buruli ulcer. We, on the NTD list, but certainly remaining neglected in many ways, given all the research gaps that are still remaining. But thank you so much for your time today. You've really helped us to shed some light on these, and hopefully we can see, just, if only from this particular project, how much of a collaborative approach you have, and lots of new ideas have been discussed today. So by all means, I hope we can all keep in touch around this, and we will be looking out for your subsequent work and further updates to all this. In the meantime, some of the attendees are joining me as well in giving you a very warm thank you, a virtual round of applause for all this. Bouke de Jong is saying, thanks to all, I've learned a lot. Roland saying, thanks for all the answers. Rachel Simmons, thanks, Koen. And as well from all of us here at ISNTD, a very big thank you for your time today, Koen, but also a very big thank you to all the attendees who tuned in to join us today. So we look forward to seeing you in future, hopefully as soon as next week for our next round of ISNTD Connect sessions. And in the meantime, we hope, we wish everyone to keep safe and all the best, and let's definitely keep in touch. So thank you very much, Koen.
Thank you, everybody. Thanks for the compliments, and have a nice evening, all of you. Cheers.
Oh, yes. I think working with us Europeans has slightly disrupted your body clock, so definitely.
I'm pretty sleepy, yes.
Yes, time for a bit of rest, and thank you. That was a big ask, coming up to midnight or 11 o'clock at night your end. So again, a huge thank you, and hope to see you very soon.
Yeah. Cheers.
Take care, everyone. Bye-bye.
Bye.
Bye.